Sutapa Datta. Welcome to my channel, Sutapa's Biology. So today I am going to explain about the most important, most magnificent the thing of cell division, that is the meiosis. Already you have studied mitosis in lower classes as well as in my last videos, and now we shall start the main thing of the cell division for class 11, that's the meiosis. So meiosis शुरू में ही बोलते हैं जो meiosis ऐसा एक डिवीजन है, एकदम फर्स्ट वीडियो में cell division 1, in cell division 1 I have told you that meiosis is the cell division which we can see only in the germ cells, the reproductive cells, not in the body cells, and not always, during the reproduction only. So meiosis, and this is the cell division 3. This is my third video of cell division. This is cell division 3. This specifically about this video I am telling you, observe it very attentively, and up to the last of the video you must observe all the videos, otherwise you will not get many informations which will be very important for the competitive exam. Now I am telling about the meiosis, and Farmer and Moore, both of them, the name of the scientists who proposed the name meiosis. This is a very important question for your competitive exam. Propose the term meiosis. Already in the previous video, that is in cell division 1, I have told you that meiosis has two divisions, successive divisional phase. Now that is the meiosis 1 and meiosis 2. And in short, I have told you also that the meiosis 1 is the reduction division. It can be expressed in this way, reduction division, and meiosis 2 is just like mitosis. So I shall describe about the meiosis 1. About the mitosis I have already given the details description in my last video. I think you have observed it. Just like the mitosis, it's the equational division. And now I am going to the meiosis 1. Now first thing is that in meiosis 1 we shall observe there the pairing of the chromosomes, and in prophase 1 it has many subphases, 5 subphases. Prophase 1, the subphases are first Deploting, then Zygote, then Kinesis. Number 1 is Deploting. Number 2, Zygote. Number 3, Kinesis. Number 4, Deploting. And number 5, Kinesis. So prophase 1 is the largest or longest subphase of the meiosis 1. Now in Deploting what happens? I am giving those diagrams. Observe very attentively. I have already told you, but this is very important for your board exam as well as for the competitive. Now in case of the Deploting, what will happen? क्या है इसमें? Deploting में क्या होगा? क्या होने वाला है, तो ये इसमें मतलब ऐसा है, this is the nucleus and the chromosome will be like this, or you can say it the cell. So here the cells are forming the thread-like chromosomes. So they are comparatively prominent than the interface, but not so prominent. So they have the appearance like the thread-like appearance in the protein. And in animal cells, sometimes you can see thread-like chromosomes. In animal cells, sometimes you can see that chromosomes, you know, the at the terminal end of the chromosome that known as telomere. Now here telomeres are converged in this manner, and the middle portion of the cell, those are projected upwards, and they are converged at one pole of the cell, and this is known as bouquet stage. So bouquet stage is the characteristics of the Deploting. Bouquet is the with the flower bouquet. It's looking like the bouquet according to the scientist, not according to me. Okay, bouquet stage. Now it's only present in case of animal cell, not in plant cell. So this is also important for you. In exam, sometimes it's given in the MCQ that where we can get the bouquet stage. Then the answer will be in Deploting. And in which cell we can see it, then it's in animal cell. You are not present in front of me, so I am giving the question and the answer, I am giving the answer also. But if you could remain in front of me, then I could ask you in this way, no? Okay, then next is zygote. In zygote, the main thing is that the cell inside the cell, there is the formation of the pair. What is the pair? Pair of the chromosomes of the same size and containing the same characters. So here in zygote, I am taking only one pair, otherwise it will waste the time if I draw all the pairs. So suppose one pair I am drawing here, and this is the centromere, very prominent now, and these two are homologous chromosome of each other. What is homologous chromosome? The chromosome which are same in appearance with each other, as well as which contain the same type of characteristics, are known as the homologous chromosome. In lower classes, I think you have studied, no? homologous chromosome. Now, one chromosome of homologous chromosome is maternal chromosome and another is paternal. We have 46 chromosomes. I think I am right. So, among this we have 46 chromosomes, and among those chromosomes, 23 we are getting from father and 23 we are getting from mother. So we are the diploid organism who have two set of chromosome, and both are homologous to each other, and they are coming from the father and mother both. And this is the main thing of the sexual reproduction. In asexual reproduction, only where only one parent is involved, then we can't get it. लेकिन सेक्सुअल रिप्रोडक्शन में हम लोगों को ये एडवांटेज है जो ऑर्गेनिज्म पापा से और मम्मा से दोनों से क्रोमोसोम लेते हैं. सो वी हैव 23 पेयर फ्रॉम फादर एंड 23 this one is father's chromosome and this one is mother's chromosome. But later you will study in my genetics video that mother has more contribution of chromosome than father. Now I am not giving the answer, as in class 12 we have the genetics, and then you will study about this, that the mother has more contribution of chromosome than father. It's not that I am a lady so I am telling in favor of mother. No, not at all. I shall give you the proof of this. Okay. So next is, in case of zygote, this is known as bivalent. Bivalent or dyad. In another word is dyad. And in this, this is the zygote is the second subphase, and the third is pachytene. In pachytene, there will be the exchange in between chromatid segments, and that's crossing over. I'm going to discuss that. But in this phase, actually in this subphase, what happens that for the crossing over, the chromatids, they should come very closer to each other that they can take part in the crossing over. Okay, so here one type of protein which is developed inside the two cells near centromere, and this is known as synaptonymal protein. This one is synaptonymal protein. This synaptonymal protein is just like the adhesive or glue, they are keeping the two chromosomes very closer to each other, binding the two chromosomes very closer to each other, and this complete configuration, that is chromosome plus synaptonymal protein, is known as synaptonymal complex. Synaptonymal complex. And the phenomenon is known as synapsis. That is, the formation of the synaptonymal complex is known as synapsis or synapse. Synapsis. So by synapsis, the paternal chromosome and maternal chromosome, they come closer to each other in close proximation. And next is pachytene. I'm going to the pachytene, rubbing all this. This is the most important subphase of the meiosis, prophase 1. Prophase 1 means it's the meiosis, because in mitosis we have studied that is the only prophase, not 1, 2. Here prophase 1 means it's the meiosis. In pachytene, this is the most important subphase of prophase 1. In pachytene we can see that the bivalent which were formed in the zygote, that's split to form a four-stranded structure. That means one chromosome is splitted to form two arms, that is two chromatids. We know that the one chromosome has two chromatids. So if the in the bivalent, the chromatids, they get separated from each other, so it will be the formation of the four-stranded structure, and that's known as tetrad. Now here two pairs of, we can see the siblings of each other, like that. If the origin is same, that is parent is same, then they are known as brother or sister, like that. So here also the same thing will happen. Firstly, I am telling about this is the tetrad. This one is tetrad, and in tetrad these two, that is two blue and two red, they are known as sister chromatid, as they are formed from the same parent. They have the same origin. Like that, one red and one blue, this is forming the non-sister chromatid. Now, the phenomenon of crossing over will take place in between two non-sister chromatids, not in between the sister chromatids. As in between the two siblings, there will be no marriage. Like that, in crossing over also, there will be no crossing over or phenomena of exchange in between the two sister chromatids. There will be the formation of the recombinant from the exchange of the two non-sister chromatids. Now here two pairs of sister chromatid, one blue pair or red pair, and two pair of non-sister chromatid, that is these two are very close to each other, so crossing over will take place in between these two. These two also, they have the same chemistry for the crossing over, but they are placed apart, so it's not possible for them to take part in the crossing over. Now what is crossing over? Crossing over क्या है? ये term क्या पता है तुम लोगों को? आप लोगों को पता है क्या crossing over क्या है? This is the exchange of the chromatid segments in between two non-sister chromatids. Exchange of chromatid segments in between two non-sister chromatid. This term is very important, that where it takes place in between two non-sister chromatid. This point is very important. Now, in the next stage of the packaging, we can see this tetrad here the synapses will be removed. In the stoppage of the synapses, as synapses has no role now, as synapses had the main role to facilitate the crossing over. It can facilitate the crossing over. So after crossing over, it has no role. Now here is the exchange of the segment, and that will be take place by the first is restriction endonuclease. This is known as genetic scalpel. This is known as genetic scalpel. As it can cut the chromatid segments. It will cut the chromatid segments at the specific site, and after this cutting, both the chromatid will be fragmented in that manner, and they will lose their track, and they will enter in the region of the next chromatid, or chromatid of the next chromosome. So this here the exchange will take place, and after that the genetic glue, that is known as DNA ligase will join these fragmented parts. DNA ligase, another enzyme, is genetic glue will rejoin these segments, and here the recombination taking place. But due to the position of the chromatids here in this packaging, the crossing over, the formation of the X-like configuration is not so clear, which we can see clearly in the deploting. So in the next phase is deploting. Next subphase is deploting. होप सो आप लोगों को अच्छा ही लग रहा है, ये वाला बहुत अच्छा है क्योंकि प्रोफेज वन इज वेरी इंटरेस्टिंग थिंग, बट आफ्टर द कंप्लीशन ऑफ दिस, अगर पूरा वीडियो होने का बाद इसका कारण हम बोलेंगे जो बहुत ही इंटरेस्टिंग है, ठीक है। तो अब भी है डिप्लॉटिंग, where the chiasma is very clear. We can see the chiasma here. Look. So in deploting, the chiasma is completely observed prominently, and in diakinesis there is the movement of the chiasma towards the chromosomal end. Chiasma formation is taking place just near to the centromere, and gradually it will move towards the end, and that's known as terminalization. And this movement is known as terminalization. At the end of the diakinesis, the chiasma will reach at the terminal part. After completion of the terminalization, this movement is known as movement of the chiasma towards the chromosomal end is known as terminalization. Terminalization, and this after this it will reach at the end, and it will be the recombinant DNA, recombinant chromosome, not DNA, recombinant chromosome will look like this manner. This is the recombinant chromosome, and these are ready for the entering the metaphase. This is recombinant, and now they are ready for the entering to the metaphase, and then metaphase, anaphase, telophase, all will be like the mitosis. In my last video, I have told you about this only the what is the difference, that the in anaphasic movement we have seen that the in mitosis the chromatid will go to the opposite pole, and here the chromosome will go to the opposite pole, and they will form from the twin N plus N. From the one twin cell, they will form the N plus N cell, and after that they will enter in the meiosis 2 for the reduction for the equational division, and there they will form 2N in one part and another 2N, that is 4N cell will be formed at the end of the meiosis, and this 4N are the gametes. It will form the either male gamete or female gamete. The next is that in between the two things, that is meiosis 1 and meiosis 2, there is the interkinesis. Just like the interphase, there is the interkinesis, but in interkinesis, there is no formation of the DNA. Like the S subphase of the interphase, there is no duplication of the DNA. Other things are same. Okay, so this is about the meiosis 1 and 2, and after that, the significance of the meiosis, that is the number 1. I have told you that it's very interesting that it's actually it can bring the variation. जो डिफरेंट टाइप का होता है, कोई एक आदमी और एक आदमी और औरत से सेम नहीं होता है, उसका कारण है, उसके पीछे जो ये है रीजन है, वो है this meiosis. So in the crossing over, the shuffling of the genes are taking place. So this is this brings the variation. Number 2, it helps in the evolution and it forms the new species. This is the main significance of the meiosis. And one thing for the competitive students, that is what is endomitosis and endodeduplication. Endomitosis. This forms the polyploids. Endomitosis forms the polyploids. Here, there is the duplication of the chromatid, but no separation. So they are forming the polyploid, and in endoduplication, there is the formation of the giant chromosome like the polytene chromosome. Okay, so this is all about the meiosis and the completion of the cell division too. So for today, bye.